Assalamu alaikum wa rahmatullah. Good afternoon to all. I am Abdul Basit from Basit Arts and Science College. I am here to present my paper on the topic Microbiology and Biotechnology Methods for Enhancing Industrial Application of Microbes and Novel Treatments. First of all, discussing about the different enzyme producing companies in our nation. We have the Enzyme India Private Limited and other different companies which all produces the essential enzymes. Mainly this paper is focusing on the microbes that can be used in the industrial level for the enzyme production, like the essential enzymes that are required for different activities in human life, like amylase, lipase and all. Okay, going forward, there are many different applications of microbes in the industrial level, as you see in my paper. The first one is the microbes of industrial valley, the heading. Under it we have the first one is that in medical industries. So the application of microbes in the medical industry is to produce a large amount of enzymes and medicines that are insufficient in humans. So it is possible to produce the enzymes and medicines in a larger quantity by different industries if we apply the microbial techniques and biotechnological events. First of all, speaking about the medical application, I have selected the penicillin. The penicillin is an antibiotic used for the treatment of bacterial diseases. This penicillin was first extracted from Penicillium notatum. First of all, combining these microbes and along with the biotechnological methods, we can use the genes present in the Penicillium notatum. Nowadays, the science world commonly use the Penicillium chrysogenum instead of Penicillium notatum. This is because the Penicillium chrysogenum, the enzyme production activity is much faster than that of the notatum one. So, moving on, we can just transfer the fungal gene that are responsible for the penicillin production, the antibiotic production, into a bacteria. That bacteria can initiate the production of the penicillin within them. This is mainly done to improve and increase the rate of production of enzymes. The researchers says that the penicillin production rate of Penicillium chrysogenum is up to 30 rate per second. But in bacteria, if the gene is inserted into the bacteria, it can do the same thing at the rate of 150. So, as you see here, this picture indicates that the penicillin gene in the black color is now transferred into the bacterial body. Here, we have a question that when a penicillin fungal gene is transferred into a bacterial gene, the fungal gene that produces an antibiotic which can kill that bacteria. So will it affect that bacteria? As a solution to this, the science introduced that to remove the penicillin binding protein. Each and every bacteria have the penicillin binding protein in their membrane, associated to their membrane proteins. So, when we remove the gene that are responsible for the production of membrane binding proteins from the bacterial body, it will result in a resistance against the penicillin, so that inserting the fungal penicillin gene into a bacterial body will not cause any effect to the bacteria that produces the penicillin. So the penicillin producing gene containing bacteria then can be introduced into a bioreactor where it absorbs the sufficient nutrition provided by the scientists who control the bioreactors. So the next one is the microbial action in enzyme production. We know that different enzymes can be produced by the microbes, which are essential for human beings and which are also essential for production of different items like bag and some fibers and all. The microbes that produces amylase. Amylase is an enzyme that is used to cleave the complex forms of carbohydrates, including starch, cellulose and all. So these complex forms of starch and all other substances are need to be cleaved in industries like food producing industries, like juice producing industries and all we have now. That all industries use this type of microbes, enzymes produced by this type of microbes to cleave the large compounds, the large complex materials of carbohydrates. Also here, the microbes that can produce the amylases are of different types, but all these bacteria that produces the amylase and the other enzyme that I have selected is the lipase. Both these microbes that produces those enzymes are not able to produce in a higher rate as the E. coli bacteria. E. coli bacteria can really increase the rate of production of enzymes. So inserting such genes into the E. coli for the increased rate of production will increase the industrial production, also can consume the time. The next one is the lipase. There are different genes responsible for the lipase production like LIMK and LIPK. The LIPK enzyme is a lipase gene that produces, that code for the lipase enzyme on the DNA of the microbe. Instead, the LIMK is a modulator gene that produces, that controls the modulation of the enzyme production, because increased production of lipase can cause dissociation of the lipid on the membrane of the cell, the microbial cell. So this modulator gene can control the rate of production, also the amount of lipase produced within the microbial cell. The next picture indicates that how to transfer a microbial gene, including bacteria, fungal or any other genes, into the required more effective bacteria E. coli. Now we can select the plasmid, the common plasmid PBR322, to insert some other microbes gene into the E. coli. Here we can see the first one. The first PBR322 is a circular plasmid which is not recombinant. In the second step, it is made recombinant by adding another gene which is required of producing the enzyme. The penicillin gene extracted from the penicillin chrysogenum is then inserted into the PBR322 using restriction enzyme and ligase. This plasmid is then transferred into the E. coli body using different transfusion methods. We have different transfusion methods like injecting directly into the nucleus or shooting methods and all. So this method of introducing the required gene into a bacteria that can effectively work on producing the enzyme fastly in the industrial level can help in consuming the time and produce a high amount of enzymes and proteins that are essential for human beings within a short period of time. So going into the next part of my paper, that is combination of this type of all the genes into a single bacteria. Just think about a company producing a lipase alone. Will it be more beneficial than producing both lipase and protease at the same time? So, if a company is looking forward to producing both lipase and enzyme, both the enzymes at the same time, they can just insert the two genes into the bacteria, into the same bacteria, and use this bacteria to replicate thousands and thousands of times along with these lipase producing and amylase producing genes. So the next challenge is to separate out these two lipase and enzyme from the bioreactor. It can be easily done using the large scale chromatography, affinity chromatography. In this picture, it is clearly indicated that in affinity chromatography, the mobile phase and the substrate phase are there. In the substrate phase, we use as the substance solid which can attract any of the enzyme, maybe lipase or amylase. If you are selecting lipase, we can just put down the substrate that have the affinity towards the lipase. So pouring this mixture of both the lipase and amylase solution into the chromatographic column, we can separate out the lipase that those stick into the substrate part. The other one, the amylase enzyme, that will come out as the eluate in the chromatography. Then the next part of my paper is the treatment using the microbial functions. This is the main part of my paper that is used for the treatment of urine in toilet wastewaters to prevent percolation of this toilet wastewater along with the urea into the well water. As we know, the Indian community or Indian society faces the main problem is that they build a house in a shorter square feet and they don't find any place to dig the well and to dig the septic tanks. So it come in very close that where the septic tank solutions or containing urea can be transmitted directly into the well water, contaminating the well water with the urea. So as a solution to it, I just got a solution for it, that is introducing the microbes that can produce urease enzyme that can cleave urea into ammonia and carbon dioxide. The urease enzyme producing microbes can be introduced directly into the septic tanks that can then cleave the urea into the ammonia and carbon dioxide. As you see in the picture, the pipe, the tube that indicates the gas outlet through which the carbon dioxide and the eliminated ammonia gas are eliminated through. The next step of this project is to consume the amount of carbon dioxide, because the eliminated carbon dioxide in the action of the action of urease along with the urea in the septic tank, the ammonia and the carbon dioxide that eliminate from this reaction is more hazardous, because the carbon dioxide, as we know, it is a greenhouse gas, and the other one, the ammonia, is a toxic gas. So the carbon dioxide can be reduced from getting into the atmosphere. It can be reduced by introducing the diatoms, which is a part of plant kingdom that can absorb large amount of carbon dioxide and use it for assimilation and just release out the oxygen as the plant does. So the next step we have is the eliminated ammonia. So how to find the solution to make it clear about the ammonia? This ammonia that are coming out of this gas outlet can be used to compress with the high pressure, which can directly reverse sublimated into a liquid form. The liquid ammonia is mainly used as a fertilizer in plants. So this converted liquid ammonia can be extracted from the container and can be used as a fertilizer. In conclusion, the microbes activity, the microbes genes and the microbes functions can be directly applied into different events of human beings' life. Thank you.
Nice presentation. That means also you